Luke Bench here from the Soils Unit DPI. Welcome to the December 2017 SNOC Soils Network of Knowledge webinar. This today on Deep Phosphorus Promotes Chickpea Root Growth by Tim Weaver from up at Narrabri. We'll have a short introduction from Graham Schwanke before Tim speaks. Graham will introduce the work on the deep phosphorus work going on up in northern New South Wales. And then we'll hand over to Tim.
Righto. Yeah, so just a brief introduction to the work and the project that the work Tim's going to talk about sort of comes under. This is part of a northern region or northern grains region project, and I guess one of the things about the northern grains region is we have some distinct soils properties with the heavy clay vertisols that predominate most of the dryland cropping area. And one of the issues, I guess, for those is, or benefits, I suppose, is that they store a lot of moisture in the soil profile during a fallow, and that can deliver a significant proportion of our annual crop yield into the next winter cropping season. And what rains we get during winter are a bonus on top of that. So we often make planting decisions based on that stored soil water. Another advantage of these soils is nutrients like phosphorus and potassium have an excellent residual value for a number of seasons after their initial application. So we've got some flexibility to apply these nutrients when cash flow and seasonal conditions suit, rather than putting them in with each crop. And I guess one of the things about putting phosphorus and potassium is that they're immobile nutrients in the soil, so where you put them is where they tend to stay. So regular applications of phosphorus into the surface layer are likely to stay there and increase availability in the surface layer, but in our dryland cropping region, quite often that surface layer can be dry for significant periods of time during the crop's growth. So we're interested in the next layer below, the subsurface layer, which is where a lot of that nutrient is extracted by. A lot of the stored water is during the crop, but with traditional fertilising in the surface, that's not being restocked as we like. So this project and several others around it are aiming at putting those fertilisers into that subsurface layer so that we can keep those crops healthy, and I guess part of the project is see how long a single application will last. As I said, it's got good residual value. So the project that Tim will be talking under today is a GRDC funded project where we're looking at both phosphorus and potassium being applied what we call deep, but when we're talking about deep, we're talking about 20 to 25 centimetres depth usually. And this particular project, as well as just looking at putting it there, we're looking at how to put it there. We've got a range of treatments such as row spacings and row orientations, and in some trials, not the one Tim's talking about, there's other methods of putting it in, whether it's strip tilling or high pressure injection or various other engineering innovations. Also, what type of product to apply. There's many different fertiliser types, and quite often when we're putting in a deep application, we might be putting in several elements at once. So we want to look in some of these trials at whether some of those elements are being tied up if you're banding them together. Also, when to apply is coming under the project as well. What's the best time of year, and finally the economics of does it pay to apply, and where in the landscape is it paying off best. So there's a whole range of things being looked at in the project. So we've set up a number of trials. The project covers quite a bit of Queensland and New South Wales, and Tim's going to be talking today on some specific data from one of the New South Wales trials, so he'll go into the detail of what the treatments are there. The treatments were set up at the beginning of last year, I believe. Anyway, he'll go into that, and we're so he's got some results from last year, and the trials have since been recropped again this year, and the plan is to keep looking at those and looking for residual value in those. So I think that's probably enough introduction about setting the scene and what it's all about. So I might hand over to Tim to give us the specifics of what he's found in that first year of deep pea with the chickpeas. Okay.
Thanks very much, Graham. And Graham's going to be available to handle for question time until he's got to leave at 10:30. So just keep that in mind if you have any specific questions for Graham. Dr. Tim Weaver, now with CSIRO, was a research officer with New South Wales DPI and is based at the Australian Cotton Research Institute at Narrabri. Tim joined the department in 1998 and has worked on a range of projects related to soil quality, soil drainage, profitability and crop rotations in crop farming systems at the Narrabri Institute. Tim has experience in minimum tillage systems and stubble retention systems in irrigated cotton farming systems. He completed a PhD in 2013 on estimating deep drainage and the movement of salt, nutrients and pesticides in vertisols. Tim's current research is focusing on exploiting the genetic, environmental and management interactions in cotton systems.
Thanks, Luke. Thanks, everyone. Thanks, Graham, and thanks for the opportunity to give the webinar today. And I guess some of the I guess part of what I'm presenting today is just a value adding to the UQ 78 deep placement of nutrient project. And I guess that stems from the cotton work that I'd been involved in very early, sort of in 2000s, where we purchased a mini risotron here at Milevale, and we used the mini risotron systems to measure root response in not only cotton but rotation crops like veg, corn, sorghum, and wheat. So when I took on this role under Graham Swanke's leadership in the project, under the national project with Mike Bell, there was just that opportunity to utilise these mini risotron tubes just to study what's happening with our root systems underneath deep placed bands. And so that's primarily what I'm presenting today, is from that UQ 78 trial at Manaroo. So I fully acknowledge here in my title page Graham and Bruce Haig and Steve Harden. So Bruce and Steve crucial. Bruce in help in assistance with the analysis of the root data, and Stephen with statistical, and Rick Graham, who is also part of the UQ 63 deep placement projects. Without the assistance of many technical and cooperators, this wouldn't happen, these projects wouldn't happen. So also I'd like to acknowledge some of the key people that assisted in taking those measurements in the field, setting up the trials across from Tamworth out to Trenny, and certainly our cooperators that allow us on to do the research work. So what I just wanted to cover briefly is just some of the complexities in the phosphorus chemistry that happens in our soils, and especially in our vertisols where we might have high calcium carbonate. You can see here a diagram that Phil Moody put together, and it just sort of explains where our different sinks of P exist and what happens in our soils. And I think on a soil test results, you always get, you can always ask for a BSES and Colwell P to determine the status of your soil profile. And what, I guess, that Zorbed P box that you can see top left is something that is a store of P that we have in our profile that seems inaccessible, but through processes it becomes available or is added to over the I guess over the seasons of adding fertiliser. And so we sometimes lose P into that Zorbed box of sorbed P that is unavailable to plant. And of course we also have other sources like organic P and the mineral P we put in, and eventually the plant's uptake through diffusion. Another test that I think I checked is around that $9 that can be added to determining your Colwell and BSES to know what you've got in terms of a bank of phosphorus as well as what is available, being the Colwell, the BSES being that which is stored, is a PBI, which is the phosphorus buffering index. And this table that I've just put up just sort of gives a range of typical PBIs, and I think you can see a range there from tenosols to ferrosols, and obviously as you move more up to those calcareosols, ferrosols, we see that they have a high PBI and of course a high ability to bind up the applied P. So, and that obviously becoming unavailable, but at the other end of the spectrum, you've got the low PBI soils, which I guess at the other extreme where you might even lose P out of the system and leaching when they're very low. So that's just another test you can ask for, and many are probably already aware of that. It sort of gives us a bit of a story and a description of what's going to happen to our applied and mineral P. And I think this next table that you'll see is one that can be used. This one I just pulled off the Birchip Cropping Group. They had this table of adjustments so that you can take your PBI and wherever that value that comes back from the lab, you can then take that PBI value and adjust your applied phosphorus to account for either if it's you're going to lose some of it to being strongly absorbed in the profile, or take into account if you're going to lose it out due to its poor holding or holding of phosphorus. So that's just a table of adjustment where if you want to raise your soil value in Colwell P by 1 milligrams per kilogram, then if you've got a PBI of say 140, then you would add 3 kilograms of phosphorus per hectare to raise that by 1 milligram per kilogram. Obviously, just multiply it by what your target for applied P that you want to achieve. You could use that table. And this is another good table which you may be aware of, that's on the GRDC website, and it's the what, where and why of soil testing in all the regions that Mike Bell and Dave Lester published back in 2015. And this sort of gives generalised critical values that we look for, that in sites where we might see some response in P. So the table shows some values of that you can look there in the surface and subsoil, and certainly addresses the availability of what you've got in store in terms of your soil test result with the BSES. So I guess if you've got values over that 60 in the surface and 100 in Colwell P, then you can see that it says unlikely to see a P deficiency in your lifetime. And you can also see that if you've got a BSES less than 25, then you've got limited evidence of residual fertiliser accumulation. So it's a good table, it's a good paper, and you can download that off the GRDC website, and it's got other nutrients as well, which you can see tables just like this in terms of critical values. So the sites that we have in the north, just to give a bit of a background of what we're looking at across where the DP sites have been established. These are some, moving from out west at Garrard, down to Gilgandra, up to Gurley, out to Ningen, and up at Taluna. You can certainly see the stratification of P. You can see that you've got some reasonable values there at Garrard for the very surface, but when you look at depth across these regions, there is just no real store of phosphorus. You can see certainly the BSES is even quite low for some of those, like at Taluna down at depth, but certainly a good store in that surface. So that's probably the key signature. I guess the key signal here is that we've got sites that are very depleted at depth. So the site that that I'll be talking about today on this map, you can see these are the sites in 2016 that were established, had been established and had crops in for looking at DP response. The site that I'll report on today is here in the Taluna region, and it's not far, I guess, east of Gurley, where we have another site west of Gurley as well. But the site that I'll report on today is this deep pea site out at Taluna. You will see now that, a photo of this season, and thanks to Graham, a drone photo of the site. So we had these tubes, mini rhizotron tubes that were placed in last year under chickpeas and this year under cereal. So to follow that up, and so the tubes that you can see there in the field and the layout is primarily how they went in the exact same plots to replicate and to follow up just the response of root systems to phosphorus. So you can see there the tubes are at an angle across the different plots where measurements were taken this year and were also taken last year in 2016 winter crop. So here's a profile of deep pea, a soil test result from the Terry Ho site. So you can see that again the stratification of the pea, where we've got around that 10 milligrams per kilogram in the surface, but drops off significantly to nothing in reserves. But again, you can see the BSES is increasing at depth, but certainly not over the 20 milligrams per kilogram at depth. Now I've just popped the potassium because there was a potassium treatment at that Terry Ho site. That's the profile in the potassium as well. So here's as Graham said, the treatments were applied in January 2016 with varying widths of deep banding, looking at if we can band not only effective banding under a plant line, but between plant lines, and if there was a reduction in horsepower and fuel usage, could we band them in alternate rows? And so you can see the tine, the Genki tine system there in the photo down the bottom. You can see here in the plots that where the deep ripping happened back in January 16, and the rates were 0, 10, 20, 40 and 80, and the treatments in which the clear tubes were placed were the 0, the 10 and the 80 kilograms per hectare plots. So that's primarily what the site looked like back in January 16 prior to the crop going in June. So here's a schematic of how the tines and plant line worked. So you can see here from left to right, this is the plant line, I guess that thicker line, and if you imagine the thinner line being the tines. So the plants were the deep banding went in, then the plants were sown straight over the same band. Where the mid row, the plant line was applied, or the deep bands were applied, and then the crop was sown between the deep bands to a depth of 200 millimeters. And then the third treatment was to say, well, if we don't need to put it in between every plant line or under the plant line, what if we went alternate and put tines in every second row, yet every plant line still has access to a deep band? And just to tease out that I guess the minimal effect of what width is necessary before we see no response from the deep bands. So the photo that should be coming up now is installing mini rhizotron tubes. So this is a diagram from far irrigated systems of how we install them. So they're installed at 45. Well, these ones were installed at 60, but I put the ones out at Terry High at 45. So just a bit more of a steeper angle, but primarily there a tube of soil is removed to allow for the clear tube to be placed in the ground, and then capped to stop daylight, UV light getting in and affecting the results. So the clear tubes are usually placed under the plant lines after they're established, so that you know you've got a good line of plants that you can be studying as the root systems go down onto the clear tubes. So up at Terry High, we hand augered those just to minimise also impact from driving tractors over the site. And what should be coming up next is a set of photos just explaining how the system, or the mini Rhizotron system, and where the tubes were placed. So you can see in the diagram you've got one tube that went through the deep band. So it's the only plot that we studied was the mid-row banding. And you can see a tube also placed on the chickpea plant line. So one on the plant line, one in the mid-row. And to the right there, you've got the computer system set up that captures the images in JPEG format. The mini Rhizotron camera, you can see on the bottom right-hand corner there. You can see, if I point that out with my mouse, the camera is actually in this little window. If you can see me pointing that out, these are UV lamps, and it also has white light lamps. So that's a mirror that's in that reflects the camera being up inside here, taking an image off the mirror from the surface. So these tubes were placed over 3 replicates in those plots at Terry High. Once the images are captured, there's different packages that you can use. This one called RouteFly was used to then quantify from a photograph to actual data that you can produce and statistically analyze. So here we've got, for example, a root system that was photographed. You can see a circle here to give us a diameter, and you can then select the root and it will scale the length of that root. And so how we get that is by taking a photograph of a 1 centimetre squared graph paper, and you trace that, and there's a calibration before you start tracing that then scales it. So every root you trace will then give you an accurate length of what you're measuring in the software. And eventually you can export all the root data that you've traced into a spreadsheet and then have it quantified for statistical analysis. So a fairly lengthy process, and takes a while. So let's get into some results from the season last year in the chickpeas. So you should see a photo now of in the root proliferation just from the initial starter. So we put 40 kgs of Granulox Z, and you can see here some of the images I pulled out from those reps in those initial stages. We were getting quite a lot of proliferation around that Granulox Z starter. So that was encouraging when I started pulling out those initial images, seeing some of those responses to phosphorus. The next image that will come up is that I've just grouped from the 0 kg plot, so they're in the mid-row band treatment. So you can see from the top left is the surface, right down, left to right, top to bottom is increasing in depth. So you can see here you've got roots that are present down through the profile, but it's sort of just single roots and not much at depth. And you can see here how the mini Rhizotron will timestamp the photos, and that means it will tell you exactly which treatment and which location down the tube the photos are taken from. So it keeps a really good record of that. So now what will come up is the 80 kilogram deep banded pea, and again in the mid-row treatment. And you can see now just how much more proliferation of the root systems in the chickpeas that has occurred, not only just in the surface, but right down deep to over 65 centimetres. So that's was really encouraging when I saw these results. And it's not only just single roots passing through each window that was photographed, it was also multiple roots of stimulants. So if I go back, I'm not sure how quick this will work, loading the two together. If I go back to that 0 kilograms, you can have a look there. compared to, now I'll flick to the 80 kilograms per hectare deep banded pea, and you can see just how much more proliferation of roots there is right down that profile. So I also just pieced together a series of photos from a 0 and an 80p, and you can see that I've just highlighted where the deep band of phosphorus was applied, and just not only it's a 75 centimetre profile of photos that have been assembled together there, but you've got the deep banding at 20 centimetres, and you can see, you know, more roots there, but you can also see that it's not only at the band, it's also pushed the root system even deeper down into the profile, which will, you know, it's encouraging to think that it will not only help with the proliferation of the root system, but also push the roots deeper to access a deeper bucket of stored soil moisture. And so that's really encouraging to see that proliferation of roots down to depth. And the next slide is now pulling that the data from photos to actual scatter plots of the root numbers that are photographed in each window. So you can see, I guess, the main point here is that you've got the rooting depth on the y-axis. On the x-axis, you've got root numbers that are present. And there's a pattern that sort of takes it from that starter. You can see certainly a lot more roots proliferating in the surface initially, and then starting to move down the profile where we've got less in the plant line, and eventually over bottom right you've got a lot of proliferation around the band, and going deeper you've got more roots counted at depth. And not only does the root numbers change, we've also can measure the root lengths, and so this is the combined sum of root lengths from each of the treatments, and FR being farmer reference, so no rip, it's not ripped, there's no tines going through that plot, and so no deep banding of phosphorus, and should replicate what's happening on outside of the trial. So you can see here that it nicely shows just how the root length does increase slowly over to November. So not too much in the initial stages, but eventually moving to the band, the roots are also increasing in length. There's more length of roots, and you can see that when those images that I showed before in the 80 kg, that if you added up all the roots in each image, the total length is going to far exceed what we saw in the 0 kgp windows. So there was a question that we also asked was, if we're seeing this increase in the root system, what about the surface? What's happening up there? And unfortunately, the crop succumbed to waterlogging disease. It was a very wet season and finished at the Terry High site, but before that happened, we did get a green seeker over the site and looked at the vegetative index, and you can see here there were some statistical differences in the mid-row and plant line between the different applied rates of deep banded pea, but no significant in the alternate row. Now, this is just one season's data, so we can't say conclusively, but it looks like the alternate row, excuse me, the alternate row is probably, the alternate row is probably not seeing any differences having the band every alternate row. But definitely the mid-band and plant line, we could see statistical differences. So I think I already had shown this photo. This is what's happened this current season, and Graham has got all the data again just to replicate and look at response under a cereal crop. And obviously, if chickpeas goes back in, it'd be good to just study and to check what we've found. It concurs with what we've found in 2016 under the chickpea crop. but this year the Terry Hi Hi site has tubes under it again in the exact same plot. So it'll be really interesting to see how those results go this year. And so I just wanted to finish off with some summary of the deep placement work. Certainly that the deep placement fee has been encouraging. It's shown that we've seen some response to the addition of the deep banded 80 kilograms of deep banded phosphorus at 20 centimetres. And also that we've seen that slow response from plant line moving across to the banding, taking a while between initial sowing and establishment through the starter to then moving out into the bands, which is really encouraging to see that it is a slow process, but we do see them getting into the band, and not only around the band, but pushing deeper. And also that we saw that root length was increasing in the presence of deep pea. And so, yeah, the last slide just to say thanks for listening. And we've got, if there's more information, contact myself or Graham Swanky and those at Tamworth as well. And also thanks to GRDC who funding the UQ 78 project. So, yeah, thank you.
Great, Tim. And I'm just unmuting Graham as well. So Kirsty Neal has written a question here, but so I'll just ask Kirsty's question, which is, hi Tim, may I ask what fertiliser types were used?
That was MAP, the deep banding P. It wasn't triple super, it was a MAP. And obviously it was also, urea was also placed to background the nitrogen, the extra nitrogen that was going with MAP, so that everything had a level playing field.
There was also a background application of potassium across all of the phosphorus plots too. So.
Yes, yeah, thanks, Graham.
Oh, Bede O'Mara. So Bede's put in a question.
Hi.
Yep, go ahead.
Hey, Bede.
How are we? Just letting everyone know, Tim, with the PBI test, mate, you get that standard with most Colwells at most labs, I believe, these days.
Excellent.
So
okay,
you don't
excellent.
need to add a PBI to your soil test request. It should come automatically with Colwell P, mate.
Yep, excellent. Oh, that's good to know. Thanks, Bede.
Thanks very much, Bede. I know Abby had one question.
Thank you, Luke. I wanted to ask Tim that given that the actual installation of the mini rhizotrons would provide a preferential pathway for root growth because they're creating a slot, does that affect your results?
Yeah, that's a good question, because when you install anything into the soil, it's always going to create that artifact beside it. And I guess the main answer is that I guess everything is treated the same. So the control will have that artifact of ease of roots growing down beside it, as well as the 80 kg. So if I guess it's an even playing field in terms of all tubes are installed in 0, 10 and the 80 kg plots, so they all have, I guess the roots all have the same opportunity to grow down in the gap that's available, that I guess isn't packed to the same bulk density as the soil in which they're growing. So that's where I sort of,
yeah, no, that makes sense. Thank you.
I'd probably add to that, Tim, in that certainly when we were putting them in this year, it was pretty tight fit. So there wasn't really much of a gap around the tubes as they were going in.
Okay.
We had to tap them in, so it was a fairly tight fit around the soil around the tubes. And I guess the other thing is that you're putting them in at 45 degrees. So while you may get some roots running, I guess, along the tube, it's more about capturing the ones as they're coming down and intersecting where the tube is underneath.
Yeah. Yep.
Yeah. Okay. Thank you.
Thanks, Abby. Well, thanks, Graham. Thanks very much, Tim, for giving our last webinar for the year. And thanks to everyone for listening in 2017 to these webinars. And on behalf of Abby and myself, I'll I'll close off there unless you had something to say, Abby.
Merry Christmas, everyone. Happy New Year, and hope to see you all next year at a SNOC webinar.
Great. Thanks all. That's it. I'll see you next year.
Thank you.